I want you to start by picturing an army. But, this isn't an army that marches over the hills or flies in on helicopters.
Right.
This is a hidden subterranean army. They are completely blind, they never sleep, and right now they're literally munching away beneath our feet.
Yeah, it's a creepy thought.
And the damage they cause to human structures, I mean, it is staggering. We are talking about an estimated $32 billion in economic damage globally every single year.
It is a massive number. And the culprits are subterranean termites. Specifically, species like coptoterms and reticuloterms.
Right.
They are just the ultimate unseen threat to our homes, businesses, our cities. Because they operate entirely in the dark, by the time you actually see the damage, the war has already been raging inside your walls for, well, months, maybe years.
Which is exactly why today's topic is so wild. Welcome to today's deep dive. We're looking at some fascinating new research out of the Journal of Economic Entomology.
Yeah, published by Oxford University Press for the Entomological Society of America.
Exactly. And the mission of this deep dive is to explore how the scientists behind this paper are asking a slightly mad scientist question. Like, how can we literally hack insect biology to speed up environmentally friendly pest control?
It's the ultimate biological arms race. And to really understand the stakes of that arms race, we have to look at the current state of termite warfare.
Which is pretty bleak for homeowners.
Very. If you discover a termite infestation in your home today, you essentially have two main choices, and neither of them is perfect. The first option is the brute force approach, which is liquid termiticides.
Okay.
These are heavy chemical barriers that pest control professionals physically pump into the soil all the way around your foundation.
And people love those because they are incredibly fast, right? Like a termite touches that chemically treated soil and it dies almost immediately, which is exactly what you want when you're lying awake at night, imagining your living room turning into sawdust.
Oh, absolutely. It offers immense immediate peace of mind. But there's a major biological catch. These liquid treatments often create what's known as a death zone. They obliterate the foraging termites that happen to wander into the chemical, but they very rarely eliminate the actual source of the problem.
The colony.
Exactly. The massive underground colony itself. You're just treating the symptom. You're killing the foot soldiers while the general is safely hidden deep underground, just breeding more soldiers.
And of course, pumping hundreds of gallons of fast-acting liquid pesticides into the soil around where we live and play carries, significant environmental baggage.
Definitely. So if you don't want to soak your yard in chemicals, you go with the second option. And this is where the new research really focuses: cheat synthesis inhibitor baits.
That are known as CSI baits.
Right. CSI baits are the sustainable, elegant alternative. Instead of trenching and pumping gallons of chemical into the ground, you place small, very targeted bait stations around the property.
These little plastic tubes in the ground.
Yeah. These stations contain a specialized insect growth regulator. This method uses a tiny fraction of the active insecticide compared to liquid barriers, and more importantly, it actually wipes out the entire underground colony, general and all.
And this is a massive, but for that stressed out homeowner, they are painfully slow. You're putting these baits in the ground and then just waiting.
Lots of waiting.
Sometimes anywhere from 45 to 90 days. In some tough field conditions, it can take up to 20 weeks to actually eliminate a colony.
Yeah.
Imagine watching a pest control guy put a plastic tube in your yard and telling you, don't worry, your house will stop being eaten sometime in the next 5 months.
It is an incredibly tough sell. 20 weeks of waiting while the termites are technically still active. I mean, people are conditioned to expect the immediate gratification of a bug spray. You spray it, the bug stops moving.
Exactly.
Handing someone a solution that takes nearly half a year to work requires a lot of patience, even if it is, vastly superior for the environment, and provides true long-term colony control.
So the million-dollar question is, how do we speed it up? Well, before we can talk about how scientists are trying to hack the system to make it faster, we really need to unpack how this incredibly slow bait actually kills them right now.
It's important context.
Because it reads like a slow-motion biological thriller.
It really does. So the process of termite baiting breaks down into 3 distinct time segments. First, there's the interception time.
Okay.
This is simply the waiting period for these blind foraging termites to accidentally bump into the bait stations while they're excavating their random underground tunnels.
They're basically just digging around in the dark, hoping they hit a jackpot of wood, and instead they hit our plastic bait station.
Right. Once they find it, we enter the second phase, which is the acquisition time.
Got it.
The foragers find the bait, they realize it's a food source, they start eating it, and they carry it back to the central nest to share it with the rest of the colony. And here is the absolute crucial part of how CSI baits function. During this entire acquisition phase, Absolutely nothing happens to the termites.
Wait, really? Nothing.
Nothing. They feel completely fine. CSIs are entirely dose independent and incredibly slow acting.
Which is by design. I mean, it's basically a biological Trojan horse. We are relying entirely on the termites' own social behaviors. The fact that they constantly groom each other and share food mouth to mouth to pull the poison deep inside the castle walls before the trap is actually sprung.
Exactly. If the bait killed them quickly, you'd end up with a pile of dead bodies right at the bait station.
Oh, and they'd figure it out.
Yeah, the other termites would immediately sense the danger, seal off that tunnel, and learn to avoid the bait entirely. The Trojan horse has to get all the way into the throne room without raising any alarms.
Right.
So the toxicant in a CSI bait only kicks in when the individual termite tries to molt.
Because itin synthesis inhibitor means it stops them from synthesizing or making chitin, and chitin is the biological building block of their exoskeleton. It's their armor.
Right. When a termite that has ingested this bait naturally attempts to shed its old skin and grow a new one, the process catastrophically fails. They physically cannot form that new exoskeleton, and they die in the attempt.
Wow.
And because of their behavioral instincts, termites naturally retreat to the safest part of the colony, the central nest, right near the eggs, the young larvae, and the king and queen, when they're feeling vulnerable and getting ready to molt.
Oh, that is so clever.
So they bring the Trojan horse into the most critical part of the fortress, and then they die.
And this is where the biology gets a little grim. The death of that single termite in the nest isn't the end of the trap. It's the spark that sets off a cascading collapse.
It's a devastating domino effect.
Yeah.
When those intoxicated workers die in the central nest, the other termites do what termites have evolved to do in a nutrient-poor environment. They cannibalize the dead bodies. Wood isn't exactly packed with protein, so they can't afford to let those nutrients go to waste.
They recycle their dead friends, which means they're just eating the poison all over again and spreading it even further into the population.
Yeah, the cannibalism accident amplifier. Suddenly, the reproductives, the king and the queen who never leave the nest, are ingesting the poison through the cannibalized food loop.
Wow.
Any eggs they lay fail to hatch. The tiny larvae, which naturally have to molt very frequently to grow, begin to die off rapidly. Eventually, the dead bodies start piling up much faster than they can possibly be cannibalized.
So the whole system breaks down.
Completely. The colony hygiene collapses, and the reproductives will actually abandon the central chamber, desperately trying to flee the cadavers.
But there is literally nowhere for them to hide. Because eventually, all the worker termites, the ones whose sole job is to forage for food and feed everyone else in the dark, die off.
Right.
And the remaining soldier termites, who have giant mandibles for fighting but physically cannot feed themselves along with the king and queen, simply starve to death.
Yep.
The castle falls completely from the inside.
It is a brilliantly effective mechanism. It takes their greatest strengths, their social structure, their nutrient recycling, their centralized nesting, and weaponizes all of it against them.
Okay, so if the Trojan horse works that perfectly, why on earth does it take up to 20 weeks for the castle to actually fall? That brings us to the real hurdle, which is the third phase of the timeline, the lethal time.
This is the immovable biological bottleneck. We can speed up how fast they find the bait by placing more stations. But the lethal time, the agonizingly long window between a termite eating the bait and actually dying, is entirely dependent on our natural molting cycle. That molting cycle is the only segment humans can realistically manipulate if we want to speed up the actual chemical kill.
And termites do not make this easy for us. They don't all shed their skin on the same day like some kind of coordinated seasonal event. They molt asynchronously. Only about 1% to 2% of a colony is ever molting on any given day.
Which means you are waiting a profoundly long time for an entire population of hundreds of thousands of insects to cycle through a molting phase.
Yeah.
And the math gets even worse when you look at which specific termites are doing the heavy lifting. The termites out looking for food, the ones actually finding your bait stations and bringing the poison home, are typically the older, mature workers.
And those older workers have incredibly long intermolt periods. An older worker might take up to 6 months to molt again.
So an older worker eats your bait, carries it back, shares it with the colony, and then just happily lives its life for another 4 or 5 months, eating your house before the poison actually triggers.
I don't get it. Look, if we know they take forever to molt, why are we sitting around waiting for their biological clock to tick? I mean, if the problem is that it takes them too long to get a lethal dose, why not just make the bait incredibly appealing?
What do you mean?
Like, slather it in whatever the termite equivalent of high fructose corn syrup is, make them gorge themselves on the poison, and speed up the acquisition time.
Well, it's a very logical instinct to want to just overwhelm their system. But it turns out the acquisition phase is already terrifyingly efficient. We don't need them to eat more. Researchers found that a single day of feeding on these baits is enough to doom an entire colony.
Wait, one day of eating is enough?
One single day. Let's look at the sheer scale of how potent this is. For a colony of about 59,000 termites, they only need to consume 1 to 4 grams of the bait matrix.
The bait matrix.
The bait matrix is just the physical material of the bait itself, usually a highly attractive cellulose powder. Within those 1 to 4 grams, there were only about 5 to 22 milligrams of the actual active chemical ingredient.
That's nothing.
Even if you scale that up to a massive mature colony of 1 million termites, they only need to eat about 15 to 18 grams of the bait matrix in total to trigger a complete collapse.
18 grams, that's the weight of a sugar packet you get at a diner. You're telling me 1 million insects only need to share a sugar packet's worth of food to wipe out the whole city?
The toxicant spreads flawlessly through their social grooming and feeding.
Yeah.
The bottleneck has absolutely nothing to do with how much they eat or how fast they eat it. The absolute immovable roadblock is that after they eat that tiny amount, we just have to sit there and wait for their internal biological clock to naturally trigger a molt. Which naturally led the scientists to the core of this new research. They asked, if we are entirely at the mercy of this biological clock, can we just hit fast forward? Can we hack their hormones so they're a force to molt right now? And that introduces us to the proposed active ingredient that has the entomology world so excited, 20-hydroxyectisone. Or, much more simply, 20E.
20E, the molting hormone.
Right. 20E is a naturally occurring hormone that acts as the trigger for the molting process in insects. The researchers' hypothesis was brilliantly simple.
Okay.
If we infuse 20E directly into the termite bait, we don't have to wait 6 months for that older worker termite to naturally decide it's time to molt. We can chemically force the issue.
So we're just feeding them the shed your skin now signal. It's like tricking their bodies into thinking it's time to change their armor before the new armor is even manufactured.
Exactly. The scientific term for what happens to them is hypericdisonism.
Whoa! Hypericdisonism. That sounds like a sci-fi weapon or a comic book villain origin story.
It essentially means they are forced into a premature, catastrophic molt that their bodies are entirely unprepared for. And in the laboratory screenings, the results were absolutely staggering.
How fast are we talking?
When researchers treated termites with 20E, using concentrations between 100 to 1,000 parts per million, the lethal time plummeted. Instead of taking months and months of agonizing waiting, the lethal time dropped to just 6 to 8 days.
6 to 8 days, that is a total game changer. I mean, that suddenly rivals the speed of those environmentally toxic liquid barrier treatments.
Yes, it does.
If you could combine this 20E hormone with a traditional CSI bait, it seemed like you'd have the holy grail of pest control. You could give panicked homeowners the lightning-fast results they're demanding while keeping all the incredible, eco-friendly, colony-wiping benefits of a targeted bait.
It looked flawless on paper. The laboratory data was undeniable. But then, the researchers took this theoretical holy grail out of the petri dish and tested it on intact, full-sized, functioning termite colonies. And, well, it completely failed.
Failed. It worked perfectly in the lab. How does it just fail?
Because real-world biology is infinitely messy. It is full of complex feedback loops, self-preservation instincts, and unintended consequences.
Okay.
When they introduced the 20E bait to a whole functioning colony, it did not accelerate the colony elimination time at all. In fact, it broke the entire baiting process. And the reason comes down to a really fascinating behavioral quirk called feeding cessation.
Feeding cessation, like they just suddenly go on a diet. What does that mean for a termite?
Think about the intense physical act of molting. A termite has to literally shed its entire outer skin. But it goes deeper than that. They also have to shed the lining of certain parts of their digestive tract.
Oh, wow.
Because of this, right before a termite naturally goes into a molt, it instinctively stops eating. It has to void its gut to prepare its internal organs for the shedding process.
Oh, wow. They have to empty the tank before they can change the armor.
Precisely. So when the researchers fed the foraging termites the 20E bait, the hormone worked exactly as it was supposed to. It triggered a premature molt almost instantly.
Oh.
But because it triggered that molt, the termites instantly felt the overwhelming biological imperative to void their guts and stop eating. The research showed that after just one single day of exposure to the 20E bait, the foraging termites experienced total feeding cessation. They stopped eating the bait entirely.
Let me make sure I'm getting this. The very mechanism that makes the bait fast-acting, the hormone that triggers the molt, is the exact same thing that breaks the Trojan horse strategy?
You've hit the nail on the head.
It's like, imagine you're hosting this massive, elegant dinner party. The guests arrive, the food is laid out. But 5 minutes into the appetizers, Someone suddenly flips on the harsh fluorescent overhead lights and starts loudly vacuuming the carpet.
Yeah,
It,
exactly.
it completely ruins the vibe. Everyone drops their forks, stops eating, and just goes home. By making them sick too fast, we stop them from acting like normal termites. They aren't going back to the nest, they aren't grooming each other, they aren't sharing food mouth to mouth. The poison never gets passed to the king and queen.
It is a brilliantly frustrating paradox. The fast-acting nature of the hormone completely isolated the affected termites from the social structure of the colony. The Trojan horse got 10 feet inside the gates, realized it had a stomachache, and just sat down. The lethal payload never made it to the throne room.
That is just wild. It is a biological roadblock that the scientists accidentally triggered themselves by being too successful at hacking the timeline. So, how do they outsmart an insect that just outsmarted our best hack? Do they just give up on the fast forward button?
Not at all. They just realized they have to get much more creative with how they choreograph the attack. The review paper proposes a few future approaches to solve this feeding cessation problem, and the most promising one is a strategy called sequential feeding.
Sequential feeding, meaning we stagger the attack so they don't see it coming.
Exactly. Since we now know we absolutely need the termites to behave normally for a little while so they share the poison, the idea is to first let them feed heavily on the standard, slow-acting CSI baits.
Clever.
You let the Trojan horse get all the way into the castle, you give them time to spread it through the cannibalism loop to the reproductives, the larvae, the soldiers, everybody.
And then once everyone in the colony is secretly carrying the slow poison,
Boom, boom. Once the CSI is widely distributed throughout the entire population, you introduce a bait with a low concentration of the 20E hormone. This hormone trigger washes through the colony, initiating the molting process all at once across the heavily intoxicated population. You get the wide, undetectable distribution of the slow bait, perfectly combined with the rapid, devastating execution of the hormone hack.
it's literally choreographing their cellular life cycles against them. First, we give them the poison, then we send the chemical signal to activate it. And 20E isn't the only hormone scientists are looking at to pull this off, right?
Far from it.
Yeah.
Researchers are also heavily looking into juvenile hormones, or JH, and synthetic JH analogs. Juvenile hormones can also induce precocious catastrophic molting. And what's truly mind-bending here is just how incredibly tiny the natural hormone levels are inside a termite. To understand how little chemical we actually need to trigger this collapse, we have to look at how they measure it.
The paper uses a term, right? Titer.
Yes, a titer in this context is just a scientific way to measure the concentration of these hormones in their bodies. The paper notes that a normal worker termite's juvenile hormone titer is just 41.3 picograms per milligram of body weight.
Let's pause on that word, a picogram. For anyone who hasn't been in a chemistry class recently, a picogram is 1/1,000,000th of a gram.
It's incredibly small.
Just to visualize that, a single grain of sand weighs millions of picograms. We are talking about an incomprehensibly small amount of material. How does something that tiny actually do anything?
That is the magic of hormones. Biological systems are incredibly sensitive to chemical signals. During a termite's natural pre-molt fasting period, that tiny 41.3 picogram amount merely doubles, and that is enough to drastically alter their behavior and physiology.
That's crazy.
Similarly, their natural molting hormones start at just 2.1 picograms and simply spike right before a molt. This tells researchers that biology is a delicate scale.
Right.
If they can just get the timing right with sequential feeding, they need microscopically low concentrations of these synthetic hormones to throw the entire system into chaos.
So the challenge isn't about brute forcing more poison into the ground. It's entirely about mastering the delivery sequence, minimizing that feeding cessation so the colony doesn't realize its biological clock has been hijacked until it's far too late.
It is a massive leap forward in how we think about sustainable urban management.
And this is why this matters to anyone listening. Why you should care about trillionths of a gram of termite hormones. If scientists can perfect this sequential feeding, if they can solve the biological puzzle of the Trojan horse, it fundamentally changes our relationship with the environment around our homes.
Absolutely.
We can finally, practically, phase out the massive liquid pesticide barriers that we pump into the soil every single year. We can replace them with these tiny, targeted, fast-acting biological hacks that use fractions of a gram of chemical to achieve total colony elimination and do it in a time frame that actually satisfies a panicked homeowner.
Moving from a sledgehammer of toxic chemicals to a highly sophisticated biological scalpel.
It is incredible science. We want to thank you for joining us on this deep dive today. We've explored the amazing tightrope that scientists have to walk between a perfect theoretical success in a controlled lab and the messy, complex reality of real-world biological defense mechanisms.
The jump from the Petri dish to the subterranean colony is always the hardest, most unpredictable step in science.
Always. And as we wrap up, I want to leave you with a final thought to ponder. For decades, the agricultural and pest control industries have spent billions of dollars worrying about insects developing chemical resistance. We constantly worry about them biologically mutating to survive our latest toxicant.
That's a huge issue.
But looking at this research, a simple behavioral instinct, the urge to stop eating to void their gut before a molt completely halted a cutting-edge hormone hack. It makes you wonder, are we entering a new era where the biggest hurdle in pest control isn't chemical resistance, but behavioral quirks?
Fascinating question.
If we start trying to hack the biological clocks of invasive species, what other hidden behavioral defense mechanisms are just waiting in the dark to trip us up? Something to think about the next time you see a tiny insect in your yard. Thanks for listening.